In this movie, we will demonstrate how to perform an SDS page and a Western blot. In SDS page and Western blotting, the proteins are first separated on a gel according to their molecular mass. This step is called SDS page. They are then blotted onto a membrane. The protein of interest is probed using an enzyme-conjugated antibody directed against the target. The antibody-protein complex is then visualized using a chemiluminescent substrate. For this demonstration, we will run a gel on reduced material, and therefore the first step is to prepare the samples by reducing the disulfide bridges using beta-mercaptoethanol and denaturating the proteins by boiling the samples. The protein is then given a uniformly negative charge according to size using SDS. First, a stock of sample diluent is made by mixing Lemley sample buffer, which contain SDS, with an equal amount of beta-mercaptoethanol. This stock is then added to the samples. Hereafter, the samples are boiled for 5 minutes to reduce disulfide bridges and to denaturate the proteins as previously described. While the samples are boiling, the running buffer is prepared, and for this experiment we use a running buffer which contains MES and SDS. We have decided to use a precast gel containing 10% polyacrylamide. However, this gel could easily be cast in the lab. The comb which separates the wells are removed, and the wells are washed using a running buffer. The gel is then placed in the electrophoresis chamber and the running buffer is added. After boiling the samples, they are then placed on ice and centrifuged shortly. The samples are then loaded onto the gel, and this is easily done using elongated tips. A marker is also pipetted into the wells on the gel. Hereafter the power is switched on and the gel runs for approximately 1.5 hours. For the blotting part of the protocol, excess gel material is first removed. First, an electrode running buffer is prepared, and then a sandwich is made, which will be demonstrated here. The sandwich is then placed in the chamber and the electrode running buffer is added. The current is applied so that the negatively charged proteins are immobilized onto the nitrocellulose membrane. After 75 minutes of blotting, the sandwich is split and the membrane is washed 3 times in wash buffer, consisting of PBS supplemented with 320 for 10 minutes. The membrane is then blocked, and for this step, various blocking regions can be used. In this setup, we use wash buffer supplemented with 5% skim milk powder. After the blocking step, the membrane is washed in wash buffer 3 times. The primary antibody is then added to the membrane and incubates overnight at 5 degrees. In this experiment, we use an HIP conjugated primary antibody, and therefore we do not include an extra step incubating with a secondary antibody. The next day, the washing step is repeated. We then wish to visualize the protein-antibody complex. For this step, we use West Pico, a chemiluminescent substrate. The substrate is prepared by mixing equal amounts of luminol enhancer and peroxidase. This mixture is then added to the membrane and allowed to incubate for 5 minutes. Excess liquid is then removed using a tissue, and the membrane is placed in the dark. The chemiluminescent signal is then captured using a CCD camera. A picture of the membrane is also acquired using an ordinary digital camera. And by overlaying these two photos, it's possible to determine if the protein is of the expected size.